receptor subunit 1, dopamine receptor subunits, calcium binding proteins. Huntington's disease. Striazomal abnormalities have been associated with the neurological disorder such as mood dysfunction in Huntington's disease. Cell types. Medium spiny neurons, they are GABAergic inhibitory neurons. 95% of total neuronal population in human striatum are MSNs, means medium spiny neurons. Primary phenotypes characteristic type are D1 type medium spiny neurons of direct pathway and a D2 type medium spiny neurons of indirect pathway. The subpopulations of medium spiny neurons contains both D1 type and D2 type receptors. About 40% medium spiny neurons expresses both DRD1 and DRD2 mRNA. Cholinergic interneurons. These secretes acetylcholine. The large spiny interneurons are affected by dopamine through dopamine receptor D5. Different types GABAergic interneurons. The best known are parvalbumin expressing interneurons, also known as fast spiking interneurons, which participate in powerful feed-forward inhibition of principal neurons. Also, there are GABAergic interneurons that express tyrosine, hydroxylase, somatostatin, nitric oxide synthase, and neuropeptide Y. Recently, two types of neuropeptide Y expressing GABAergic interneurons have been described in detail, one of which translates synchronous activity of cholinergic interneurons into inhibition of principal neurons. These neurons of the striatum are not distributed evenly. There are two regions of neurogenesis in the brain, the subventricular zones in the lateral ventricles and the dentate gyrus. Neuroblasts that form in the lateral ventricular adjacent to the striatum integrate in the striatum. This has been noted in the human striatum following an ischemic stroke. Injury caused to the striatum stimulates the migration of neuroblasts from the subventricular zone to the striatum, where they differentiate into adult neurons. The normal passage of SVZ neuroblasts to the olfactory bulb, but this traffic is diverted to the striatum after an ischemic stroke. However, only a few of the new developed neurons survive. Inputs. The largest connection is from the cortex in terms of cell axons. Many parts of the neocortex innervates the dorsal striatum. The cortical pyramidal neurons from layer 2nd and 4 sends their projections to the striatum. The most dense projection comes from layer 5. They end mainly on the dendritic spines of the spiny neurons. They are glutamatergic, exciting the striatal neurons. Ventral striatum receives many inputs from the cerebral cortex and limbic structures such as amygdala, thalamus, and hippocampus, as well as from entorhinal cortex and inferior temporal gyrus. Another well-known afferent is the nigrostriatal connection arising from the neurons of the substantia nigra pars compacta. While cortical axons synapse mainly on spine heads of spiny neurons, nigral axons synapse mainly on spine shafts. This afferent is glutamatergic. The participation of truly intralaminar neurons is much more limited. The striatum also receives afferent from other elements of the basal ganglia such as the subthalamic nucleus, glutamatergic, or the external globus pallidus, GABAergic. Now we can see here the scheme of input and output from the cortex to the striatum, and from striatum to the substantia nigra, from globus pallidus to pars externa, pars interna. Here when we ventrolateral thalamus, then again cortex, and with this you can understand by giving proper time. Here let's go ahead. Targets: the primary output of the ventral striatum goes to ventral pallidum, then the medial dorsal nucleus of the thalamus, which is a part of frontostriatal circuits. Additionally, the ventral striatum projects to the globus pallidus and substantia nigra pars reticulata. Other projections includes to the amygdala, to the lateral hypothalamus, and to the pedunculopontine nucleus. Striatal outputs from both dorsal and ventral striatum consists of median striatal neurons, a type of projection neurons. Basal ganglia core. Basal ganglia core is made of striatum along with the neurons, which gets direct projections via the striatopallidonegral bundle. The striatopallidonegral bundle is a very dense bundle of sparsely myelinated axons, giving it whitish appearance. The projection comprises successively the external globus pallidus, the internal globus pallidus, the pars compacta of the substantia nigra, pars reticulata of the substantia nigra. The neurons of these projections are inhibited by the GABAergic synapses from the dorsal striatum. Among these targets, the external globus pallidus does not send axons outside the system. Others send axon to the superior colliculus. Two others comprise the output to the thalamus, forming two separate channels, one through the internal segment of the globus pallidus to the ventral oralis nucleus of the thalamus, and from there to the cortical supplementary motor area, and another through the substantia nigra to the ventral anterior nuclei of the thalamus, and from there to the frontal cortex and the oculomotor cortex. On the picture, overview of the main circuits of the basal ganglia. The striatum is known in blue. Here we can see picture show two cortical slices that have been superimposed to include the involved basal ganglia structure. Plus and minus signs at the point of the arrow indicates respectively whether the pathway is excitatory or inhibitory. In effect, green arrows refer to the excitatory glutamatergic pathway, and red arrows refers to the inhibitory GABAergic pathways, and turquoise arrows refer to dopaminergic pathway that are excitatory on the direct pathway and inhibitory on the indirect pathway. Functions of the striatum. The ventral striatum, mainly nucleus accumbens, mediates reward cognition, reinforcement, and motivational salience. The dorsal striatum mediates cognition involving motor functions, certain executive functions, i.e. inhibitory control, and stimulus response learning. Dorsal striatum is also a part of the reward system along with the nucleus accumbens core, mediating the encoding of the new motor programs associated with future reward acquisition. For example, conditioned motor response to the reward cue. Metabotrophic, G protein coupled. Dopamine receptors are present both on spiny neurons and on cortical axon terminals. Second messenger cascades triggered by activation of these dopamine receptors can modulate pre and post synaptic function both in the short term and in the long term. In humans, the striatum is activated by stimuli associated with reward, but also by aversive, novel, unexpected, or intense stimuli, and cues associated with such events. fMRI evidence suggests that the common property linking these stimuli to which the striatum is reacting is salience under the conditions of presentation. A number of other brain areas and circuits are also related to reward, such as frontal area, Functional maps of the striatum reveals interactions with widely distributed regions of the cerebral cortex, important to a diverse range of functions. Clinical significance of striatum. Parkinson's disease. Parkinson's disease results in loss of dopaminergic innervations to the dorsal striatum and other basal ganglia, and a cascade of consequences. Atrophy of the striatum is also involved in Huntington's disease, chorias, chorioathetosis, and dyskinesias. Addiction. Addiction, a disorder of the brain's reward system, arises through the overexpression of the FOSB, a transcription factor in the D1 type medium spiny neurons of the ventral striatum. FOSB is a inducible gene which is increasingly expressed in the nucleus accumbens as a result of repeatedly overdosing on an addictive drugs or overexposure to other addictive stimuli. Bipolar disorders. There is seen to be an association between striatal expression of variants of the PDE10A gene and some bipolar disorders. First patient, variants of other genes, DISC1 and GNAS, have been associated with type 2 bipolar disorder. Dysfunction. Dysfunction in the ventral striatum can lead to a variety of disorders, most notably depression and obsessive compulsive disorder. Because of its involvement in the reward pathway, the ventral striatum has also been implicated in playing a critical role in addiction. It has been well established that the ventral striatum is strongly involved in mediating the reinforcing effects of drugs, especially stimulants, through dopaminergic stimulations. Pallidum. The pallidum consists of a large structure called the globus pallidus, pale globe, together with a smaller ventral extension called the ventral pallidum. The globus pallidus appears as a single neural mass, but can be divided into two functionally distinct parts called the internal or medial and external or lateral segments, abbreviated as GPI and GPE. I for internal, E for external. Both segments contain primarily GABAergic neurons, which therefore have inhibitory effects on their targets. The two segments participate in distinct neural circuits. The GPE receives input mainly from the striatum and projects to the subthalamic nucleus. The GPI receives signals from the striatum via the direct and indirect pathways. Pallidal neurons operate using a disinhibition principle. These neurons fire at steady high rates in the absence of inputs, and signals from the striatum cause them to pause and reduce their rate of firing. Because pallidal neurons themselves have inhibitory effects on their targets, the net effect of striatal input to the pallidum is a reduction of the tonic inhibition exerted by pallidal cells on their targets. Disinhibition. This is called disinhibition, with an increased rate of firing in the targets. That's all. Thank you.